All right. Welcome back, everyone, to the deep dive. We're diving into something pretty fascinating. Today, it's all about epigenetic aging.
Okay.
So, you know how we always talk about DNA as this blueprint, right? It's got the instructions for life.
Right.
Well, what if I told you there's like another layer on top of that blueprint, kind of like instructions for the instructions? That's where epigenetics comes in.
Yeah.
And today, we're focusing on a key player in that, DNA methylation.
Okay.
But before we get too deep, got to remind you guys, please don't forget to like the video and subscribe if you want more content like this.
Right. So yeah, DNA methylation is like super important for how our genes actually get used.
Right.
Basically, it involves adding these little chemical tags called methyl groups to one of the DNA building blocks, specifically cytosine, especially when it's next to a guanine.
So that's the CPG sites we hear so much about.
Exactly. CPG sites.
Yeah.
And this whole process, it's crucial during when we're developing.
Right. Makes sense.
But what's really cool is how these patterns change as we age.
Okay, so how does the body actually put these methyl groups on there?
Good question.
And keep track of them over time?
So we've got a whole set of enzymes dedicated to this. First up are DNMT3A and DNMT3B.
Okay.
You can think of them as the architects laying down the methylation blueprint during development.
Got it.
Then when cells divide, DNMT1 comes in along with this helper protein, UHRF1. They're the maintenance crew, making sure the methylation patterns are copied onto new DNA strands.
So making sure the blueprint stays consistent.
Yeah, exactly. But it's not a one-way street. We also have TET enzymes, TET1, TET2, and TET3 that can actually remove those methyl tags.
So it's constantly being adjusted, a dynamic process.
Exactly. It's a constant balancing act.
Fascinating. So, but we've talked about these methylation changes with aging, but how do scientists actually track them?
Well, that's where aging clocks come in.
Aging clocks, that sounds kind of ominous.
Not really. It's actually quite clever. Researchers figured out that if you look at how methylation changes at specific CPG sites across different ages.
Okay, across a whole bunch of people.
Yeah, you can actually build models that predict not only your chronological age.
Like how many birthdays you've had.
Right, but also your biological age, which is more about how well your tissues and organs are actually functioning.
Oh, wow. So that's really getting into how old your body really is, not just by the calendar.
Exactly. And these clocks, they usually rely on what we call bulk sequencing data. It gives an average picture of methylation across a group of cells.
So that's looking at individual sites, how they change as we age. But the research we're looking at today, they did something different, right? Like an orthogonal view.
Yeah, you got it. Instead of just seeing if methylation goes up or down at one site, this research looked at how pairs of sites change together.
Pairs of sites.
Yeah. So it's not just about one site, it's about how they're connected and how that relationship evolves as we age.
Okay, so this is where it gets really interesting for me. We're going beyond looking at these individual marks and seeing if they're talking to each other as we age. What was the big goal of this deep dive into coevolution of methylation? What were they hoping to find?
Well, they really wanted to understand these collective changes in DNA methylation during aging. Looking at how these pairs of sites influence each other over time, they hope to uncover some fundamental mechanisms driving the aging process itself.
Makes sense.
And they analyzed a huge amount of data from different mouse tissues.
So they looked at different parts of the mouse, not just one specific area.
Exactly. Looking for consistent patterns in this coordinated change.
Okay, so did they find evidence of these coordinated shifts?
They did. The big finding was that as the mice aged, DNA methylation at sites about 500 base pairs apart became more correlated.
Wait, so more likely to be the same? Both methylated or both not?
Exactly. You can think of it like two light switches that start off random, but over time, they're more likely to be in the same state, both on or both off.
That's a great way to picture it. But why 500 base pairs? Is there something special about that distance?
Absolutely. That distance is right around the spacing between a CPG site and a CPG island and the edge of that island, what we call the shore.
Okay, backing up a sec. CPG islands, those are the areas with a bunch of CPG sites, right?
Yeah, they're often near the beginning of genes and usually unmethylated, which lets those genes be active.
Okay, so these correlated changes are happening specifically at the edges of these regions near genes.
That's right.
So it's not just random. It's tied to these regulatory features of our DNA.
Exactly.
So these shores are kind of like the transition zones around genes.
Yeah.
Why are they important?
Well, they're more dynamic than the center of CPG islands. Methylation changes more readily there, and that can subtly influence the activity of nearby genes. The fact that the correlation changes specifically at these shores suggests that, well, it suggests that regulation of those transition zones gets messed up with age.
Ah, that makes sense.
And to back this up, they looked at areas with fewer CPG sites, what we call CPG poor regions, and found that the correlation stayed the same with age, no matter the distance.
Interesting. So it really seems specific to CPG islands and those shore regions.
Right. They even saw that the distance where this correlation increased actually seemed to scale with the size of the CPG island itself. So the boundaries are definitely key.
Okay, so it's definitely not random. It's connected to these specific regulatory features. But how do they explain this increasing coordination? What's the mechanism?
They came up with this really cool biophysical model.
Okay.
And it suggests this whole thing comes from a shift in the balance between the enzymes that add methylation, like DNMT3A, which is more active around CPG islands, and the enzymes that remove it, the TET enzymes, which are usually more active within the islands.
So it's like a tug of war between adding and removing methylation.
Exactly. And as we age, that balance shifts a bit at these shores, causing what they call a wetting-like phenomenon.
Wetting-like. I don't quite follow.
So think of a water droplet on a surface.
Oh.
If the surface changes, the water might spread out more. Here, it's like the influence of one site's methylation on its neighbor gets stronger with age, specifically at those shores, making them more likely to be the same.
Ah, okay. So it's not that the methylation itself is spreading, but the influence of one site on another is getting stronger.
That's a good way to put it.
But what's actually causing this shift? Like at the molecular level, what's going on in our cells?
They think a key player might be PRC2, the polycomb repressive complex 2.
Okay, I've heard of that. It's involved in silencing genes, right?
Yeah, and it's linked to a histone modification called H3K27Me3. And there's some evidence that PRC2 levels, or maybe how it interacts with DNA, actually decrease as we age.
Interesting. So if this PRC2 complex is becoming less common, how does that affect methylation correlation at these CPG island shores?
Well, PRC2 and its H3K27E3 mark probably influence both DNMT3A and TE activity.
Okay.
You see, both H3K4ME3, which is an activating mark often found in the middle of CPG islands, and H3K27E3 can actually block DNMT3A from binding.
So they both prevent methylation from being added.
Right, and at what we call bivalent CPG islands, which have both activating and silencing marks, both H3K4ME3 in the center and H3K27E3 at the shores.
So they're like ready to go either way.
Exactly, they're poised. And the reduction of PRC2 and H3K27E3 might disrupt that balance with the activating H3K4ME3. This could then change DNMT3A and TE activity specifically at those shore regions.
So it's like as PRC2 declines, that balance gets thrown off, affecting the methylation equilibrium at those boundaries.
You've got it.
Okay, that makes sense. But what evidence actually links this to PRC2?
Well, for one, the increased methylation correlation with age was strongest at these bivalent CPG islands, which we know are targets of PRC2 components like EZH2 and SUZ12.
Okay, that makes sense.
And they also saw that the width of the H3K4ME3 peaks at these bivalent islands actually got wider with age.
So the activating mark is spreading.
Yeah, and that could further mess with the methylation machinery in those nearby star regions.
So it's like this cascade.
Yeah.
Less PRC2, more spreading of the activating mark, and then that throws off the balance between adding and removing methylation at the edges of these CPG islands, leading to this increased coordination.
Yeah, exactly. That's a great summary. And what this all suggests is that aging isn't just random damage accumulating, it's a more organized shift in how our epigenome is set up. That's pretty profound.
Yeah, it really changes how we think about aging. So for our listeners out there, you're always eager to learn new things quickly. This research gives us a whole new way to understand aging. It's not just about isolated changes. It's about how different parts of our epigenome are working together and how those relationships change over time.
Right, and it raises a huge question. If this coordinated methylation is such a big part of aging, could we target those changes to, maybe even influence the aging process?
That's a huge question. It could change how we think about anti-aging therapies.
Absolutely. It opens up a whole new world of possibilities.
Wow. This has been another amazing deep dive. Thanks so much for being here today.
My pleasure. It was great to talk about this.
And to all you amazing viewers, don't forget to like the video and subscribe if you want more deep dives like this.